Hi everyone! My video today is about 4 laboratory practicals focusing on the method of culturing the feed for fish larvae. In this video, I will explain about microalgae culture, counting microalgae, rotifer culture and microworm culture. Microalgae culture. Firstly, we need to pour the starter culture into a container. Then, use the syringe and add 5 ml of fertilizer into the container. After that, add salt mixture of 1.5 salinity into the container of starter culture. Lastly, turn on the air pump and your microalgae culture are ready. Counting microalgae. For making Lugos solution, we need to dissolve 50 gram potassium iodide and 25 gram iodine in 100 ml boiling water. Then, dissolve 25 gram sodium acetate in 250 ml water. Mix them in one beaker. Next is counting cell in a sample. Take a 5 ml of microalgae sample, add 1 drop of Lugos solution and mix well in a test tube. Put 1 drop of well-mixed algal suspension on each Neubauer chamber and cover with its cover slip. Now, place the Neubauer chamber on light microscope. Start counting at the top left square and count only those cells which lie within or touch the boundary line chosen according one of the two possibilities in rotifer culture. The rigid tubing was cut into 3 lengths of 12-inch and 4 lengths of flexible tubing. Rigid tubing was inserted into culture bottle, air pump was turned on. Flow rate was adjusted on the splitter as necessary to create uniform moderate flow. Few ounces of phytoplanktons were added. Make sure aeration is on and the culture will watch over each of the next few days. When the water clears to a light green tea color tint and a little speck was seen in the water when the bottle will hold up to a bright light, a few ounces more phytoplankton was added. After 2 days that the rotifers have cleared the phytoplankton, second culture was started. 0.5 liter of the culture of the teal color liquid was taken and was poured into a second plastic bottle. To harvest the rotifers, 3/4 of the teal color culture was poured out of the culture through a rotifer sieve and into a discard water change bucket. After the culture through the rotifer sieve had filtered, the sieve was turned upside down over second bucket. A cupful of the discarded culture water was scooped out and was poured slowly over the upside down rotifer sieve, effectively reversing the flow of water, and the collected rotifer was rinsed into the collection bucket or trash can. Microworm culture. Cut the potato into half. Make a hole inside the potato. Dig a hole and place the cut potato. After a week, take back the potato. To start the microworm culture, get a clean container and make a hole at the cover using needle. Place the oatmeal into a clean container. Mix the water and oatmeal together. Take some starter from the oat culture and put it on the oatmeal. Put some yeast in the culture and see the result after a few weeks. In conclusion, for a good growth of the larvae, we need to provide enough protein and energy. This will increase the demand and profit in the marketing. Other than that, some of the live feed was easily to culture economically and low cost than artificial diets. A disease-free healthy stock can be maintained by feeding live food to the culture stock along with the supplemented artificial feed. A disease-free healthy stock can be maintained by feeding live food to the culture stock along with the supplemented artificial feed. Supplemented artificial feed cannot meet all the elements required for the growth of fish, so fish and shellfish must be fed with live food. Thank you for your attention.